you for taking the time to view this video poster presentation. My name is Silvin Knight and I'm a postdoctoral research fellow at Trinity College Dublin. Today I'm going to present our work titled Blood Pressure Entropy as a Novel Marker of Frailty, Initial Results from the FRAILMatics Clinical Cohort. This work is taking place as part of the Science Foundation Ireland funded FRAILMatics research programme. The overarching goal of this project is to develop tools to detect subtle signs of frailty and new models of multisystem dysregulation to stressors. This is being achieved through the development of such models using a large population-level cohort, and these results are being validated using a smaller clinical cohort. And it is the first of such clinical validations I'm presenting today. Although frailty status is often categorized into distinct groups, as is the case with the widely used Fried frailty phenotype, it is actually a continuum ranging from robustness to severe dependency and death, and can be thought of as a marker for biological aging. Other frailty instruments, such as the Frailty Index proposed by Ken Rockwood, or the Share Frailty Instrument, better take the continuous nature of frailty into account. Entropy is the measure of the amount of randomness or predictability in a closed system, with higher entropy being indicative of greater randomness or unpredictability. Entropy can be used to quantify the amount of disorder in continuously measured physiological signals, and in this study we used sample entropy for this purpose. We have previously demonstrated that higher signal entropy in resting state neurocardiovascular data is associated with worse pre-disability frailty status, worse cognitive performance both cross-sectionally and longitudinally, accelerated brain aging, and greater risk of all-cause mortality. Entropy levels in blood pressure signals could provide a clinically useful marker for systemic cardiovascular dysregulation, and by extension worse health outcomes. However, work to date has been performed using the TILDA dataset, a large population-level cohort, and validation on a smaller clinical cohort of patients is needed to test its clinical utility. With this in mind, the aim of the present study was to extend our previous work and investigate whether entropy in blood pressure data was associated with frailty status in a clinical setting. In this study, we used the Share Frailty instrument to quantify frailty status. Although this approach provides a continuous variable, it approximates Fried's frailty definition by using 5 similar variables, namely loss of appetite, fatigue, grip strength, physical activity, and functional difficulties. In this study, we used data from a cohort of patients over 50 recruited from the Falls and Syncope Unit at Mercer's Institute for Successful Aging in St. James's Hospital. Data was acquired continuously during at least 5 minutes of supine rest using a Phenopress Nova device, and the last minute was used in the entropy analysis. All entropy analysis was performed in MATLAB, and optimal M and OR variables, which are inputs into the entropy algorithm, were derived via hyperparameter tuning. Statistical analysis was performed in Stata, and 3 models were investigated. Model 1, a univariate approach. Model 2 includes basic demographics and health measures as control variables. And Model 3 extends Model 2 to also include lifestyle and socioeconomic factors. In total, the cohort consisted of 100 patients, with a mean age of 70 years, and 55% were female. These plots show results from the hyperparameter tuning. This approach revealed that in order to maximize the beta coefficient and minimize the p-value, an M of 5 and OR of 0.4 were optimal for diastolic blood pressure models. Our study revealed a significant linear association between higher diastolic sample entropy values and higher ShareFi score, as illustrated here in the margin plots. This association held for all 3 models investigated. There was no significant association between systolic blood pressure entropy and frailty score. Our study demonstrates that blood pressure signal entropy could provide a clinically useful indicator of the physiological dysregulation indicative of worse frailty status. The methodologies developed herein were specifically designed to be easily transferable for use in the clinic, requiring only resting state data, which is relatively quick and easy to measure, being computationally fast, and as such could potentially provide an at-the-bedside measure. The measure is easy to interpret and track over time, since it's just a single value, and it's also quantitative and an objective approach. Thank you for your time.